Welcome to our channel Forensic Genesis. The topic for this lecture is biological development of fingerprints. As we have already covered the introduction and history of it, the content that we'll covering in this particular lecture is structure of skin, formation of layers, and morphogenesis of friction ridges. Let's start with the structure of skin. The skin consists of two layers, epidermis and dermis. Epidermis is the outermost layer of the skin and dermis is present beneath the epidermis, which is 15 to 40 times thicker than the epidermis and constitutes the primary mass of skin. Beneath the dermis, there is a layer hypodermis, or we can say subcutaneous fatty tissue. If we talk about epidermis is further divided into 5 sublayers. The first one, if we start from the bottom, is basal generating layer, or we can say stratum germinativum or stratum basal layer. The second one is spinous layer or stratum spinosum, then granular layer or stratum granulosum. Fourth layer is transitional hyaline layer or stratum lucidum. The fifth one, the topmost layer, is horny conified layer or stratum corneum. Now let's see the formation of these layers. So the formation starts from the granular layer, which is the conified layer. It forms skin cells called keratinocytes from the granules of keratin. These skin cells finally become the conified layer, that is the stratum corneum, the outermost epidermal layer. This layer exposed to the environment made up of 15 to 20 layers of flat dead cell, which sheds regularly through abrasion or replaced by keratinization. As you have noticed on your hand, your skin peels normally in so many scenarios. So that is the abrasion or peeling of skin, the first outer layer of the skin. So all the cells originate from initial cuboidal shaped cells formed on the basal layer, that is above the lamina, that migrated through the epidermal layer up to the conified layer. Cell move upward with the surrounding cells. Basal cells do not migrate, it remain attached to the generating layer, that is dermal papilla. Now what is dermal papilla? It is basically a part of uppermost layer of the dermis, which is made up of mesenchymal cells. We can say it is the bottom layer of epidermis where this generating layer is present. All these cells are forming over here. This is like a finger-like projection, increasing the surface area between the epidermis and dermis, thereby strengthening the juncture with the epidermis and increasing the amount exchange of oxygen, nutrients, etc. All during this process, cell changes shape, reduce activity, and take up keratin, that is the water repellent protein. All cells of the epidermis therefore originate from the basal layer and the interstice between the dermis and the epidermis, that is the dermal papilla, where the original formation of these friction ridge, these layers are happening and cells are moving upward accordingly. So the permanency of friction-rich pattern is largely due to this generative process, whereby the cell constituting the epidermis are produced on the inner protected basal layer just above the dermis. Only damage to the basal layer will result in permanent scars in the epidermis. Now let's just see the biological development of fingerprints. So the morphogenesis of friction ridges, how this is happening and where it started from the first time. So it starts in the first week of the gestation period. Hand starts to develop within 5th to 6th week of the gestational period. Then the development of first finger is between 6 to 7 weeks. Then there is appearance of volar pads between 7 to 8 weeks. As you can see in the diagram, there is a formation, or you can say the appearance of volar pad, which is basically a swelling of mesenchymal tissue under epidermis on the volar surface of the fetus. So these volar pads appear on each finger at 7 to 8 week and remain clearly visible until 10th week, when growth of hand overtake the pads, rendering them not visible by week 16, as it is clearly visible in the diagram also. By the week of 16, it is invisible to us. So this whole process or phenomena is called as regression. So the major development of friction ridge takes place between 11 to 20th week. After 18th week of EGA, or estimated gestational age, friction ridge system stop adding new ridges. The ridges are locked into their configuration. So whatever ridge formed over there is locked, and there is no adding of any other new ridge. By the week of 24th in estimated gestational age, the formation gets completed. That was the process of formation of friction ridges. Over there I mentioned morphogenesis, that means development. So basically it is development of friction ridges. Friction ridges is the raised portion of the epidermis. So the formation of pattern reflects the size and shape of volar at the time of formation of friction ridges. So if we talk about the whorl pattern, it is formed on very high and rounded volar pad. If we talk about loop, it forms on the asymmetrical volar pad that leans to one side. And in case of arch, the volar pads are very low, thus results in the arch pattern. Thank you for watching. If you like the video, please like, subscribe and share.